They are indole compounds. Indole 3-acetic acid or IAA is the most common auxin. Auxins are produced in the shoot and root apical meristems or tips of a plant. They are also found in the immature leaves and seeds. Auxins promote stem elongation. They also promote apical dominance. Apical dominance occurs when the shoot apex inhibits the growth of lateral buds so that the plant may grow vertically. This kind of growth will help the plant to get more light to conduct photosynthesis. In this plant, there is an active apical bud, and it is in this bud the auxins are produced. Even though auxins promote stem elongation, they inhibit the growth of the lateral buds. As a result, the lateral buds will not grow into side shoots. Now, if I remove the apical bud, the auxin concentration lowers, and because of this, the lateral buds will grow into side shoots. Auxins promote the activity of vascular cambium. Vascular cambium produces secondary xylem as well as phloem and result in the thickening of stem. Auxins help in root initiation. They also help in fruit growth. As I explained in the case of apical dominance, auxins inhibit lateral bud growth. They also inhibit the abscission, or in other words, dropping of leaves and fruits. Now have a look at these Arabidopsis plants. Arabidopsis plants are used in various plant research studies. Here is a normal Arabidopsis plant with normal levels of auxin. But this genetically modified plant cannot produce auxin. See the difference in growth shown by these two plants. So from this, we can conclude that auxins are promoters of plant growth. Now let's see what are the functions of cytokinins. They are adenine derivatives. Creatine is the most abundant cytokinin. Cytokinins are produced mainly in root tips. Small amounts of cytokinins are also produced in actively growing plant parts such as developing shoot buds, young fruits, etc. Cytokinins are called so because they promote cytokinesis, or in other words, cell division. They also promote cell differentiation, that is, specialization of cells. They play a major role in promoting shoot growth. Cytokinins also promote opening of stomata and chloroplast development, thereby promote photosynthesis rate. They also cause movement of nutrients to tissues where their concentration is higher. They inhibit apical dominance caused by auxin, which means that they help the lateral buds grow into side shoots. They delay leaf senescence. Leaf senescence means aging and gradual death of leaves. Here, these two tobacco plants were grown under drought conditions for the same period of time. The plant that has less number of green leaves is a normal plant. There is no surprise that its leaves have become dry because that's how a plant reacts to a drought condition. The plant with lots of healthy green leaves is a genetically modified plant. The specialty of this plant is that during drought stress, it can produce more cytokinins, and since cytokinins reduce aging of leaves, the leaves are greener and healthier than the normal plant. However, cytokinins inhibit root growth. But overall, cytokinins are promoters of plant growth. Now, what about gibberellins? They are terpenoids. Gibberellic acid 3 or GA3 is the best known. Gibberellins got their name because they were first isolated from a fungus, Gibberella fugicuroi. Gibberellins are produced mainly in developing seeds, also in shoot and root tips and young leaves. One of the main functions of gibberellins is that they promote seed germination. They help in mobilization of food stored in the endosperm to the growing embryo. Another function is promoting internodal elongation. Internode means the part of the stem between two nodes. Can you identify these tall plants in this picture? Then be ready for a shock. They are cabbage plants. Yes, cabbage plants treated with gibberellins. These two cabbage plants are the normal ones. In the gibberellin treated cabbage plants, you can see the internodal elongation caused by gibberellins compared to the normal cabbage plants. Gibberellins promote flower formation and promote the production of male flowers in the place of female flowers in plants like cucurbits. They also promote pollen tube growth and fruit development. Gibberellins inhibit seed dormancy. Overall, gibberellins are also promoters of plant growth. Next phytohormone is ethylene, which is the only gaseous hormone. Of all the phytohormones, ethylene has the simplest chemical structure. It is produced during seed germination, ripening of fruits, abscission of leaves, and senescence of flowers. Ethylene promotes aging and senescence of plant organs. It causes leaf epinasty or drooping of leaves. In this picture, among these two tomato plants, the one on the right side was exposed to ethylene. After some time, the leaves showed epinasty or drooping down of leaves. Ethylene also promotes fruit ripening. One of the main functions of ethylene is promoting abscission of leaves, flowers and fruits. Ethylene is the primary and most important chemical involved in the process of abscission. It inhibits stem elongation. Overall, ethylene is a growth inhibitor. Next phytohormone is abscisic acid or ABA. They are carotenoid derivatives produced in the mature leaves, dormant buds and seeds. The name abscisic acid was given because researchers at first believed that it was this hormone that stimulated abscission or falling down of plant parts. Today, this name abscisic acid is a little misleading because it is now proven that even though abscisic acid promotes abscission, it does not play a primary role. As I said earlier, it is ethylene which plays the primary role in abscission. Abscisic acid promotes dormancy in seeds and buds. It also promotes closure of stomata under drought conditions so that less water is lost through stomata and helps the plant survive in such unfavorable conditions. In this picture, these two plants were under drought stress for the same period of time. Among these two plants, the plant treated with more abscisic acid analog shows better tolerance towards drought. As I mentioned earlier, together with ethylene, ABA promotes abscission of leaves. Abscisic acid is a general inhibitor of plant growth. It also inhibits seed germination. So overall, abscisic acid is an inhibitor of plant growth. Even though when I say that ethylene and ABA are growth inhibitors, under certain unfavorable conditions like drought stress, it is necessary to slow down or inhibit the plant growth to make sure that the plant uses less water and survive the drought conditions. Because under drought, the strategy is to survive rather than grow. So, growth inhibitors are also actually good and necessary for the plant growth and survival.